Hi everyone, welcome to this webinar. I'm really excited to have all of you joining us today. I'm sure that all of you have heard about CRISPR, and it's really taken off over the last several years and has spread through different areas of biology, enabling all sorts of research and application that would have been very difficult. And today we have Dr. Sahil Gupta with us, who will give a talk about step-by-step guide of using CRISPR technology for primary cell genetic engineering. So I want to start by introducing you our wonderful speaker today. Dr. Sahil Gupta finished his PhD in cancer immunology at Goethe University of Frankfurt, and then he did a postdoc at European Research Institute for the Biology of Aging at University Medical Center Groningen in the Netherlands. And then he founded his first company, ProLevi Bio, that works on revolutionizing the treatment for hypothyroidism, which is an autoimmune disorder. And then he has more than 10 years of experience in the field of immunology and genetic engineering. And today in this webinar, he will be sharing step-by-step guide for successful genetic engineering with latest CRISPR technology development. And you can find today's agenda already in the chat box. And throughout this event, if you run into any technical issues, please send a message into the chat box. And also, if you have any questions or anything that you want to share with our speaker, you can also send in the chat. And also, please note that I'm recording this event and will share this recording for others who could not join us today, so they may also benefit from the content. Okay, and now I welcome Dr. Sahil Gupta. And if you are ready, you can start. Yeah.
Thanks a lot, Yenyo. I guess you can hear me. And
yeah, we can hear you.
Good. Perfect. So thank you, first of all, for that amazing introduction. Today I'll be talking about how to efficiently target basically your homologic construct in primary cells using genetic engineering CRISPR-Cas9 technology. And this experience I mostly gained, during my time in postdoc. And I would like to share this with you today. So the agenda of the meeting is it's already shared. We'll first talk about different classes of Cas cleavage enzymes, right? The modifications, for instance, and epigenetic activators, base editors. We'll then move on to specific modifications that will help you increase the HDR efficiency in your primary cells. I've listed kind of a history of that. I will go into that, and most of these techniques or the case studies I've listed, I have experience with. And finally, I'll be sharing some summary of these technical protocols towards the end. And if you need something really specific, you can always reach out, and I will share full protocols with you as well. So very briefly, how does this single guide RNA assemble? There is a target sequence, which is called CRISPR RNA, a 20 base pair sequence, and trans-CRISPR, trans-activating RNA that basically supports this scaffold. This forms a complex with Cas9, the protein, which is the cleavage protein, right? And this entire complex is called ribonucleotide protein complex. If your target is specific, there will be a cleavage just 3 base pairs upstream of a site, which is called PAM site, so proto-spacer adjacent motif, right? And then the outcome would be, in most likely scenario, non-homology mediated end joining, basically NHEJ. This will lead to something called indels, so basically insertions and deletions in most of the cases. With a very brief history of how this basically was evolved, this is a bacteriophage, so virus immune mechanism against, so basically bacterial immune mechanism against viral infections. The bacteria basically collects the particles of DNA from the virus and incorporates them into some spacer units called repeat spacer units, where this name comes from as well. These fragments are read as basically barcodes after incorporation. And once this is transcribed, this forms a complex with what we call trans-CRISPR RNA, right, to form the scaffold with Cas9 protein. So when there's a next infection, right, this spacer element will specifically recognize the alien sequence from the virus and target it. So like I mentioned earlier, the phenotype or basically of a cleavage or a damage in your DNA is either non-homology end-mediated joining, which leads to indel, right? Or in rare cases, HDR, right? So homology-directed repair. Now this, to optimize the HDR, is what will be the focus of today's talk. Briefly, with different classes of Cas enzymes, so cluster-associated, short repeat elements. It's divided into two classes, largely class 1 and class 2. Class 1 includes mostly archaic bacterial, Cas enzymes that are involved in bacteriophage immune response. And Cas2 are mostly, a DNA or RNA targeting that can be exploited, especially in the mammalian cells. Starting with Cas9, it has two nuclease domains. So basically, it will cut the enzyme either on the top strand or the bottom strand. And this is called as RUVC or NHH domain. It recognizes a PAM sequence, which means you need an NGG sequence. In this particular case, which is GC rich, followed by a 20 base pair specific complementary sequence to your DNA. The substrate is double-stranded DNA. And it leads to blunt ends. The next one is type 5A, so Cas12. And this also has the two nucleus domains. The target is double-stranded DNA as well. And for Cas12, this is also, with the new version of enzymes, RNA, right, the target. It leads staggered ends. And the PAM sequence recognizes basically AT rich. Then you have Cas12b, which only has one domain, right? Which means that if it binds, it will only cleave the non-targeting strand. So RUVC basically gives the non-targeting strand. The substrate is also, double-stranded DNA. And then there's Cas13a, and there have been, tremendous updates in the field. However, with this current talk, we will only talk mostly on Cas9 nucleases. So the first modification I would like to talk about is Cas nickase, right? And this you can use for deletion in HDR. This is ideal for targeted deletion for up to 100 base pairs or oligo insertion for up to 200 base pairs. So this is basically, the catalytic enzyme domain of the Cas9. I will not go into the details, but like I said, so RUVC1 cleaves the targeting strand, and HNH H840 cleaves the non-targeting strand. And if both of these mutations are present, it will basically catalytically inactive Cas9. So Cas9 D210A nickase inactivates basically RUVC domain and cleaves the target strand. So if this is basically your guide RNA targeting the strand, and this is the mutation, here is the cleavage. Basically, that will happen where the complementarity takes place. Cas9 H2840A has mutation in HNH domain, which is the other domain. So basically, it will cleave the opposite strand. Now, there are two configurations if you want to insert your oligo, basically PAM out and PAM in. PAM out means that your single guide RNA has NGG on the opposite ends, right? So 3' end here and 3' end here. And PAM in means that the 5' end are basically, towards the middle. The most ideal scenario, as adopted from this AdGene website, is PAM out. So basically, if you can spread your guide RNAs for targeted deletion. The most optimal spread of guide RNA spacing in this scenario, if you're using the D210A version, right, which is the RUVC, is 37 to 68 base pair of spacing, right? Efficiency in this regard, would be up to 80%. If you go beyond, it will be less. H840A, right, has relatively less, basically, separation, but sorry, a little bit more separation, but less efficiency. Therefore, the most efficient configuration, in my opinion, is the D210A with the RUVC domain, right? Separated 37 to 68 base pair with a PAM out configuration, right? So try to spread it out if you want target deletion. Now, if you want to use it for oligo insertion, right, you will have to follow a certain, few more steps. Apart from the basic PAM out configuration and, choosing, and I would go again for RUVC, D210A, you had to place your homology arm in between like 40 to 100 base pair range, right? You can also do perform basically unequal homology arms, so that increases the efficiency. And always when you order an oligo, right, from ITT, order both basically the top and the bottom oligo to test this configuration out. There are also lots of other modifications of this oligo, but we'll talk about it later in the next part. The second modification is basically if you want to activate your promoter region, right, or a certain enhancer region even. And we'll be talking about 3 such activators, which is VP4, SAM, and SUNTAG, and inhibitors, so basically epigenetic silencers, or such as KRAB and a recent versions of epigenetic silencers as well. So very briefly, the idea is if you fuse, any, let's say, a moiety to your Cas9 protein, or your even single guide RNA moiety, right? You can recruit exogenous factors that you can use for epigenetic activation, silencing, recruiting other cofactors, also for gene tagging, right? And also imaging, and the applications are basically, tremendous regarding that. Now, the first version was VP64, which is activation cofactor, basically, and recruits, P300 and cofactor domains for activation. This was the first version that came out, VPR, followed by SAM, right? And I really like this version. And this version basically co-recruits other cofactors to increase the efficiency, such as MCP, P65, and HSF1. And the way it recruits is by adding, recruitment domains to MS2. So MS2 is basically your guide RNA scaffold, which is modified. And then there is SUNTAG, so which basically incorporates peptide motifs, right, that you can visualize by, expressing certain antibody tags. So basically your entire genome will light up. What I've tried back in the day was the first version. So this is the add gene numbers for these, and this you'll get at the end as well. And it has basically EF1 alpha promoter with DCAS9, so catalytically inactive VP64, and the MS2 loop, so basically that you will need for cloning your guide RNA as well. The loop, like I said, basically recruits, coactivation factors such as MS2, P65, and HSF1. And this increases the efficiency compared to just the original version of VP64 alone. If you have just MS2, this already increases the efficiency, but it increases much higher, right? If you add a further tetra loop moieties that are binding moieties for all of these cofactors. This I regard as the second version of, effectiveness. Then there are other attempts made as well from George Church's lab, and this is just one of them, where they further tried to, truncate these regions and try to incorporate it further, basically, into the Cas9 backbone. There they find this efficiency, which is the mini version of VP64, to be 36%, 36 times more efficient at certain target genes, and up to 24 times, at certain other target genes. These are the add gene numbers for this version. I recommend that you try it. I have not tried it, but the strategy definitely looks quite promising. There are other methods as well, which is basically incorporating this entire thing in one polycistronic, gene region, such that you express all of these factors into one. And this current version is basically lower in size. We'll talk about it later as well, why the size of your delivery vector would influence the efficiency of editing. Because in primary cells, it is really difficult to get huge bulky vectors in. Compared to the size that exists, like of the previous version, 10 KB, this is a truncated version, right? Around 5 KB, so easier to get inside the primary cells as well. Then there is also the Suntag version, like I said. So for this, you will need to incorporate and basically clone in some peptide epitopes, knock into your protein of interest that you want to basically track, right, for imaging. And then you can exogenously express this peptide moiety, right, which will bind basically and light your protein of interest if one of your aim is, let's say, tracking this protein in long-term imaging, or also just normal imaging. The next version basically that is the repressive version, and this is the first version that I used back in the day with KRAB. So KRAB is basically a repressive domain, right? So it recruits repressors and basically silences your target gene of interest. What you see here is I used basically this guide RNA to target the enhancer region, which is 7.5 to 8 KB upstream in primary human macrophages. What you see is basically the response of your CCL18 gene, right, which is just the target gene, with your just control vector, right, and with basically the repressing targeting vector. So basically, if you block the binding sites of STAT6, right, or even STAT3, in this enhancer region, the overall expression of your gene for CCL18 is decreased. With IL-4 and IL-4/6, this is increased, and after basically just blocking this region, this is further decreased. This was confirmed basically both through just qPCR and chromatin immunoprecipitation, so basically another technique, chip assay. The delivery vector I used for this was BSmart, so you can use this as well. Now there's a better versions now for silencing now available. The last I checked, there were like, I think 5 to 6, more publications last year and few from last month. But in my opinion, the next possible thing that you should try, or the version you should try, is the one with a ZIM3 repressive domain. The reason for that is the authors tried and compiled 57 KRAB repressive domains, and they came out with ZIM3 basically as one of the most effective one. But do know this is just done in hex, and it's quite recent publication. It's not validated in primary cells. But the way it works is they screened in a reporter assay, and the single guide RNA that you use for targeting was a TET repeat. So basically, the more fusions you have, right, of this 57 KRAB domains, the more silencing would take place, and this is what you will see as a GFP readout. And this is what they validated. And this version is available on Agene as well, so you can also order this. The next is base editors, right? This is for single nucleotide substitutions. And for this, the first that was introduced was in 2016, which are cytosine or adenine base editors. The way they work is how they work is they have a deaminase domain, which is linked to a nucleotide that they will recognize. This nucleotide, they will deaminate, which means they will take the amino acid out of that, such that a cytosine will be converted to thymidine and a guanine to alanine, right? So this C will be deaminated, right, to target C. This will be read as uridine, and then this will be then converted to thymidine. So basically, your C converts to T, and the opposite is also true for G2A. And of course, the same for adenosine base editors, only the target nucleotide changes. The enzyme, of course, is different in this case, which is activation-induced deaminase, right? This is an ortholog from a bacterial gene. The concept is the same, like I said. And the only caveat is that with the older version, you cannot really predict the efficiency quite reliably, right? Which is why it didn't get huge traction. However, before we get into the more recent versions, what are base editors and how they work? They have Cas9 nickase, so basically only one enzyme site is active. Like I said, C converts to U in a 5 nucleotide window, which is created in this bubble of Cas9. The UGI, basically the uracil glycosylase inhibitor in this enzyme, inhibits this excision, such as this uracil is read and then basically converted to thymidine. So there's a nickase activity to make the non-edited strand, and this leads to directed cellular DNA repair process, with the G-containing DNA strand. However, it was noted that cytosine-based editors, are highly ineffective. So research now is mostly focused on adenosine-based editors, and there are some really good modifications. The next version was adenosine-based editors, second version, where you would appreciate that the corrected base edit now from A to T to G to C is up to 48%, right? The concept is pretty much the same, apart from uracil. This is, inosine, right? And that is then converted to guanine at the end, adenine to guanine and thymine to cytosine. So what modification did they perform? They firstly tried in different, deaminases available right from E. coli to human, but they failed also. But then in E. coli, they tried with this ETAD DCAS9 domain, and they found one region that basically rescues, this mutation, which is this D1008 to N mutation in this particular case in the amino acid. And this mutation basically rescues the chloramphenicol gene phenotype that they were screening for resistance to. The third generation basically has further modification where they include ApoBEC deaminase enzyme, which is more specific. And I apologize for the bad image quality. And you can also either use this in trans, right? Which means, with an exogenous reporter vector with mCherry, or in cis, right, with mCherry or GFP, with the BE4 version, or with a version with a cleavable GFP, so expressed, just from one basically plasmid. The advantage of this is basically, and the newer version, which incorporates basically XCAS9 and SPCAS9NG, which is this recent version, is that it expands basically your NGG target site, and this can be really hard in case you want to look for base substitutions. This has lower off-target effects, and it's more DNA specific. For human version, mammalian version, this is called CRISPR-Cas, right? And authors have now basically compared all of these, right? Comparing the AB7 second version, the version I showed you 2 slides ago, and the most recent version, basically. I'm sorry, are there any questions?
No, I think somebody had.
All right. And just to show that basically the efficiency per se, in terms of the reporter that the authors evaluated, increases up to 25%. So if you really want to use this version, I would recommend using this latest one with DCAS9 and AB Max, which is the 4th generation version. Now, how do you increase your modifications? How do you basically increase your HDR efficiency with CRISPR modifications? I'll start with a brief history, and the initial attempts that were made were basically at inhibiting NHEJ, right? If you inhibit basically indel, the logic was, you would increase HDR. Or if you activate any pathways, let's say that promotes HDR, the idea was, you would indeed also increase your efficiency. This was published basically, 2016, 2018. and this is called crispy mix, right? This version that in one small molecule inhibits basically NHEJ, the other tricostatin, right, and MLN 4924 and NSC 11520 increase the HDR basically in these versions. And indeed, the authors basically, if you compare these columns that targeted nucleotide substitution, right? If you just focus on the green, they kind of increase, with 2 to 3 percentage points. I mean, needless to say that this is not ideal, because if you're using primary cells and you start inhibiting these, pathways, it's quite dangerous in terms of off-target effects, and especially with immortalized cell lines, because you might generate, lots of non-specific clones. So I would not recommend that, but if you really want to do it, I would probably go for, for immortalized cells and not primary cells. The second strategy was cell synchronization, and there's a very good reason for that. But before I go into that, the strategy in this case that the authors used was they modified their HDR template, right? So they used the wild type version of Cas9 and guide RNA, but the HDR template they added a restriction site, hint 3. You can add this restriction site for further validation, right? If your knock-in is efficient or not, but do make sure that this is outside your coding region, right? So in basically either homology arms that is not in the coding region, because obviously you might end up including another amino acid that you don't want. Nevertheless, the strategy used was to synchronize the cell cycle, and the idea was to increase such efficiency. Again, I would not recommend it, but it is an option, for increasing efficiency, especially with nocadizole, right? Because this brings your cell cycle into arrest, at least partially at G2, and synchronizes and releases such that most of the cells are released in S phase in time, where the most HDR efficiency takes place. Why? Because this is the phase where your homology directed, repair is the most active, basically in S to G phase. And with that idea in notion, the authors, conjugated your Cas9 or coupled Cas9 with 2 enzyme geminin, right, which is active, for HDR during this phase, and CDT1, which is mostly active throughout the cell cycle, for non-homology and joining. And with geminin alone, right, what you ensure is that your Cas9 is only active in the phases that is required, where the HDR actually takes place. So you minimize the off-target effects. And this is also demonstrated basically in this Western blot, where you see that with geminin in G0 and G phase, there is very less or low levels of Cas9, whereas in S and G2 there are high levels of Cas9. Whereas with CDT1, it doesn't really, change much, because the levels stay throughout. So if you really want to ensure that, you want less off targets, I would go for probably the geminin version. The next is basically coupling the tagging or protection of your oligos with something called biotin and phosphothiourate protection for your HDR template. Before we go into that, so the authors used in this particular study something called Cas9 monoevidin, which you will also hear in this talk. This is Cas9, which is coupled to a monoevidin, so basically a protein conjugate. And if you couple your oligo with the biotin, this will form a bond with this monoevidin and ensure that the oligo is only close to your Cas9. So it only is really targeted and intended where you want your oligo to be. And this coupling you do basically, in vitro before the start of the experiment. So you make sure that only your oligo is conjugated to Cas9. They also use something called pi, right? This is a really, robust and highly, cheap method it ensures that your plasmid remains inside the cell for up to 4 weeks. I would not recommend it for primary cells because of the sheer off-target effects that it might have. But for immortalized, if you have budget issues, please give Pi a shot. It acts like Fugene or Jet Prime, right? It's a version based on liposome complex, right? So I would recommend to use it if off-target is not an issue and if you're working on immortalized cells. Nevertheless, like I said, so this biotin would conjugate with that you have on the oligo, on the streptobiotin tagged Cas9 protein. And this also has basically a spacer, right? A spacer region. Another method where this was successfully used was something called of a phosphorothioate bond. So this basically is the phosphate backbone oxygen replaced by sulfur. What it does is it prevents your oligo against the action of endonucleases, which is the enzymes inside the cell that would basically cleave your oligos. What the authors did was they cloned basically their HDR construct, right, with the GFP tag, and they added not just the phosphorothioate bond, but also the biotin bond. So basically it's 5' biotin with phosphorothioate bond. And this you can order, as a primer directly from IDT, right? It's super cheap. And this ensures really that your oligo stays in close proximity and ensures good efficiency for a primary edit. You can even go up to 2 kb version, right? If you amplify your HDR for this oligo with the Cas9 monoavidin and biotin and phosphorothioate bond conjugation method. I've tried it successfully and it does work in primary cells, but I'll talk about it later. There's another method called PCV-Cas9. This is the porcine circovirus 2 rep protein. It has binding efficiency for a 13 base pair region and also a certain kind of cleavage efficiency with the HUH domain. So if you conjugate your single-stranded DNA with this 13 base pair region, this will bind to this protein, right, the PCV protein, that is then conjugated to the Cas9, will form the complex with single guide RNA together called the RNP, plus your oligo is also already conjugated to this, PCV-Cas9, because this assembly takes place basically inside a tube. And then you can either transfect, electroporate, whatever you want inside the cell. Once the HDR takes place, only the oligo is inserted. So there's no, extra nucleotides. If the homology is successful, that will be inserted. And the same is also true if you had any doubts regarding, the previous strategy. So only the specific homology arm is inserted and nothing, no scar tissue. Sorry, no scar DNA. This PCV binds covalently to the single-stranded oligo, and this is basically what the author, showed. They used basically a reporter system, right, where they used to repair the M-cherry, SNP. And what you can see after successful insertion is both M-cherry and GFP are being coded. Make sure that you use PCV-Cas9, which is N-terminal tagged Cas9, and on the Cas9 PCV because it has a lower efficiency. The next study is quite brilliant, to be honest, and this was published, I think, Poonam Malik's group. What they did was they catalytically inactivated the binding domain for P53, right? And they started mutating this domain, right? They label this domain basically as DN1S domain. So what this domain does is it just competes with the full length 53BP1, such that this protein complex cannot come and bind to your double-stranded break, because if it does, it will induce apoptosis. And if it does, your viability of cell decreases. The second thing it does is it also increases the efficiency, because right now you do not have to non-specifically block, all these pathways. Only specific block at the site of insertion transiently just for 24 hours, right? So this is the version that I would really recommend that you use as well, the DN4, version. Here's the add gene link basically for this plasmid. They show this basically also inside the cells, mostly HEC, but also a bit primary cells, but they're immortalized. Nevertheless, the efficiency definitely, increases in this regard, either with, SP-Cas9 or also the other version of SA-Cas9. Here they also show how the HDR efficiency increases and how basically your overall indel, decreases compared to just, if you would use the standard approach. The next one is a case study where they used efficient scarless genome editing. So there's a two-step process. It's a long process. It's what they do basically in the first step is that you have your one guide RNA that actually goes and binds and inserts your big insert, right, with a promoter, M-cherry construct, and also a plasma membrane localized, protein. In this case, let's say CD19, right? You screen specifically with flow cytometry through the CD19 because you can push it out, and also with M-cherry you can screen. This is step one, right? In step two, you use a second guide RNA that you inserted in this cassette, and you give the homology arms around your actual edit, the one that you really wanted, right? The loss of signal, right, from this M-cherry and CD19 will help you then pick up the clones, right? So it's a two-step process, but the timeline is longer than 3 months as they stated. However, it's really quite an elegant approach if you want to try it, truly scarless approach. All right, last one is my favorite. This is prime editing. I tried this in primary melanocytes. I'll spend some time here. So the idea of this technique is that you have a reverse transcription domain, right? That tags your, let's say that has your 13 base pair of inserts that you want to incorporate. And then basically it uses a few base pairs, right, from the genome as a starting template and starts amplifying this region such that it's really specific. Okay, how this technique works. How this works is that you have basically your DNA, right, that you want to target. Then you have your the loop, right, which you called the PEG RNA loop. This has a single guide RNA binding domain, right, which you call spacer. In the previous slides, we talked about this. And then you have basically the other arm of this domain, right, that includes a primer binding site, which is 13 base pair site, highly spec, which is complementary to the first 13 base pair, basically, of your spacer region, right? And then you have your reverse transcription template site, where you actually have your edit, right, that you really want to edit. So these 3 base pairs. You can also include further silent mutations, right, beyond the 7 base pairs. Yeah. So the 3 base pairs need to have your edit, and then your 4 base pairs, which with actual complementarity. And after that, you can basically extend with further, silent mutations that will help, really get targeted insertion, and such that your guide RNA does not cleave your already edited region. This then leads to polymerization of this edit, right, at the target site. And then there are like 2 scenarios, right? Either there is flap equilibrium and your 3' flap in this region, has your edit, and it's basically, cleaved out, or sorry, it's incorporated in with flap equilibrium, or your 5' flap has the edit, right? In this case, you will not get basically the edited version, right? And if basically, it's in the 3' flap region, you get the edited DNA version. So this is how it looks like. Basically, you have your spacer region that binds to the DNA. Then you have your, hair loop region that is synonymous to trans-CRISPR RNA. Then you have your reverse transcription template, right? The 15 base pairs 13 base pairs we talked about. And then you have your primer binding site, which is exactly complementary to your binding region of interest. So the authors tried basically this in quite a few scenarios in HEX as well, with the first version of the enzyme, right? This is MML-RT. And this was really not efficient version. So this is called PE1 version. The next version were actually PE3 and PE3B. They're all coded by the same, plasmid. The only difference is how many additional nucleotide you add, right? Or how far you are from the nick. And that basically classifies the terminology, right, for this. So like I said, there are a few possibilities. There's a possibility that, after the addition and peg RNA encoding of, let's say, your, edit, there's 5' excision, and then, ligation. In this case, the sequence remains unchanged. I think I misspoke earlier. So with 3' flapping excision, it remains unchanged. But with 5' flap excision, right, and using PE3 and PE3B version, you will get your preferred nick repair, and then this will be, changed to the complementarity version of your actual edit. As you can see, with the 2 version, the efficiency and correct edit is not that high. It's still like 15%, which is quite high, but not high. But with 3 and 3B version, this increases to 40 to 50%. Below you see another example in a different gene. And in this particular case, with a nick that is 48 base pair, downstream of your edit, right? This version, you get basically higher efficiency. So then you will need, not just the PEG RNA version, but also a Cas9 nicking version, right? A single guide RNA nick that basically nicks around, around 40 to 50 base pairs from your actual PAM site, right? What this does is basically creates a loop such that this reaction can actually take place. So you really have to use another guide RNA for the nick. The final case study is regarding the transposon encoded CRISPR-Cas system. This is really a landmark study, and it is still under development, only possible currently in the bacterial versions, right, not mammalian. So the idea and why this is important is because transposons have a huge cargo loading capacity, right? More than around 10 kb and even higher, with highly specific and efficient insertion if you are successful. With the current versions, the limitation is 2 kb, 5 kb, and it's quite cumbersome, especially in primary cells. So the authors did attempt this in the cholera strain, where they successfully inserted this cargo. The way this works is that this is coupled Cas system with your transposon system, where the transposon cargo basically has your DNA loop, right, or your insertion site, the HDR template that is incorporated up once the single guide RNA recruits this entire machinery to your target strand. So basically what you have is a Cas version that recruits basically your single guide RNA and the transposon domain to the target site. And then as soon as it's recruited, there's a cleavage 49 base pairs downstream of your PAM site, right? And this cargo is pretty much inserted inside the target of interest. Like I said, only possible in non-mammalian systems yet, but the same is being developed. This is basically, again, a representation of what vectors they used if you are interested in performing it, or reading a bit more into that. But now came out basically just actually a few months ago, a paper on transposon mediated RNA programming in mammalian system, but this is very close to as you can get in non-mammalian system. This is very close to as you can get in mammalian system. And what I would recommend is to keep an eye out on this particular group or publication, because this will be the next biggest thing in terms of targeting of your HDR templates. Now, just to briefly summarize of exactly how to use these techniques, which cell types to use, and which version of Cas9 to use, what protections that you should do. This is what this final section of next 2 to 3 slides would cover. The first one is basically, if you want to use the p53DN1S system that locally blocks your p53 recruitment and increases the efficiency, I would recommend the lentiviral insertion or electroporation, starting with 200,000 cells. We did contact the authors for a purification system, for purification of Cas9DN1S system. And this would really increase if you can purify the Cas9 system. So do reach out to Punapalek, the lead author of this study. And if you can get this version, that would be the most ideal and specific in this regard. The next version is Cas9 monoevident. For this, you have to protect your oligos with biotin and phosphothiazolate bond. I would go, with lipofectamine-based systems or electroporation, mostly if you're talking about primary cells. That should be a go-to strategy. Homology arms depends if you're just using oligos, of course, just 100 base pairs, right? On either end, 40, 60. And if you're using big HDR constructs and you want, let's say, a GFP inserted as well, I can go up to, you can go up to 2 kb with homology arms around, 600 base pairs. And unequal homology arms actually help, right? It's not a bad thing. With the RNP concentrations, you have to test it out, right? If you're using the plasmid version, with the polycistronic non-primary cells, use around 1 microgram for a million cells. But if you're using really for primary cells in RNP version, I would always start, with a lower concentration of RNP and do a test gradient, right? Lower is actually better in most cases because it does not induce a huge toxic effect in your cells, and the cell largely do not die, and the efficiency actually increases. The next version is the Geminin version, right? Like I said earlier, if you get your hands on a version where you can transcribe this Geminin, right, with a T7 promoter, that's better than the plasmid version, I would say, for primary cells, of course. And these are the conditions if you want to use this transfection system or electroporation system from the authors. The next version is, let's say, if you do not want to use, the Cas9 monoevident version, but you just want to go with wild type Cas9, that is also okay, right? But in this case, I would still recommend that you protect your oligos, right, and do a co-transfection. The oligos will not bind with the Cas9, version of your protein, or in this case, Cas9 mRNA version, right? But you have to protect your oligos from the endonuclease activity within the cell. It's mostly ideal and recommended, this strategy, if you want to do like, in vivo or in zebrafish. The next, like I already mentioned in the first attempt, if you want to go with just pure oligo approach, it is possible, right? Make sure you insert some, restriction enzymes inside, and you also protect your oligos, right? It is kind of important if you want to go with this version. And the PCV Cas9. Unfortunately, this version did not work for me, and the reason for that was mostly technical, because I think a purification of the protein was not 100%. But this is really a promising technique. And also, all you have to do is just order a simple primer with 13 base pair attachment. If you have some biochemistry, capabilities within the team or in your vicinity, you should try it out. So what has worked for me in hand, like I said in past, are these conditions. For primary human macrophages, I use the RNP complex in 2:1 ratio in monocytes. I let them recover for 3 days. You differentiate them basically, for further few days, and the efficiency is quite high. For knockout at least 60%, and for silencing, the epigenetic silencing, I saw a silencing effect of around 30 to 40%, and I think that's with the version 1, right, back in 2015, and now there are better versions. Like I said, the ZPR version, with CareApp. So please try that out. That will work. Then I've tried it also in primary cells, in mouse B cell precursors. These are the conditions I use. Do note I use very low, concentrations, and you can even go lower depending on the cell numbers you use. That's really important. I got an efficiency for knockouts, for up to 70 to 80% in this scenario. For melanocytes, primary human melanocytes, I tried 2 approaches. I tried multiple, but the 2 that worked was using a wild type single guide RNA Cas9 RNP complex with a plasmid-based approach, right? I used a reporter, GFP, that helps you in just initial screening, but after 4 weeks, the plasmid will basically be shunted off, right? So it won't be expressed because it's not incorporated. This plasmid is just SDR plasmid, a cloning plasmid, not insertion plasmid, right? But remember, you will have to evaluate, let's say, if you want to include, certain markers or not. I did, it worked, but you really have to make sure that your efficiency is high for insertion. And you can compromise the viability on that if you increase the transfection efficiency, right, which is assessed just 48 hours after transfection. The second that worked for me was the single guide RNA Cas9 monoavidin and tagging of oligos for a base edit in primary melanocytes. So finally, what do I recommend right from start until the end? Test 3 to 4 single guide RNA around for your knock-in of interest, right? Use tide analysis. All of this documentation is available with protocols, towards the end if you just reach out to me. And start with ribonucleotide proteins for primary cells, starting at 20 to 120 micromolar ratio. So basically 20 with a 2 to 1, so single guide RNA to Cas9, 20, 40, right? 30, 60, up to 0.1 1 million of cells, and assess really, truly your single guide RNA, efficiency. This second, you can do already with plasmid-based guide RNA approach, right? And once you've validated your plasmid-based guide RNAs, you can then order, synthetic guide RNAs, right? I generally order it from IDT with CRISPR RNA that you can couple with the TRUS, CRISPR activating RNA and the ATO550 dye. With the right flow cytometry technique, you can also kind of assess this quantitatively in terms of what your transfection efficiency was. Then finally, basically optimize your transfection efficiency with a GFP reporter or of similar size, right? If you plan to use HDR plasmid, like I did in this setting. Then for co-transfection, I would always, it's recommended to also use a smaller plasmid from this publication, around 3 kb, because it increases the efficiency of your insertion. You should really aim for a high transfection efficiency, right? Electroporation efficiency, whatever, at a low viability cost. But you will have to find the right balance for that, right? For point mutation, I recommend that you clone your template in a cloning plasmid, which is generally PUC19, which is a one-step, cloning technique, at least using infusion HD cloning, and always incorporate silent mutations because you do not want your guide RNA to go back and recognize the same, spacer region and cleave again, right? So try to mutate your PAM, try to mutate, the regions within your silent mutation regions within your spacer region. Now, if you're using oligos, like I recommended already, protect your oligos, right? Irrespective of if you're using the monoevidin version or not. Actually, the biotin that binds with monoevidin. Include silent mutations, and you can also include restriction enzyme sites, because what you can do is that you can amplify this region and just do a cheap 30-minute restriction enzyme assay, and just assess it, if it worked or not on PCR. And the only caveat is that this is not that sensitive of a technique compared to something like a droplet PCR or the other qPCR if you want to assess it. So the effect you might only see at 5 weeks. Make sure, please, that the restriction enzyme is outside your coding region, right? You don't want to screw up any reading frame. Then design primers basically outside your HDR template for validation, right? And one set of primer you should always have with one arm, which is at the silent mutation, at your HDR, basically, oligo. This will help you, identify the true edit. The first step would be that you amplify from outside the arms, right? Just send it for normal Sanger sequencing and use, again, a few tools to assess your HDR efficiency before you spend, more time in characterizing if the HDR actually, take place, or even keeping your cells in culture, for 1 month. So with that note, I would like to end the talk very briefly. All of this work that I showed is stemmed from, the past experiences, right from a PhD that I started in 2014, finished in 2018. My experiences at, UMCG, where I did my postdoc at Areba, funded by the DFG fellowship as well. My time at the iPSC CRISPR Center, and finally, currently, I am now working on an autoimmune disorder, like was introduced earlier, where we really try to help patients, suffering from this disease, and find a proper cure. And with that note, I'm happy to take any questions that you have.
All right. Okay, yeah, thanks really a lot for this very informative talk. That's like a very, very nice overview about all these different CRISPR developments and also the applications what people can do with this great technology. And so if we have questions from the audience, you can now unmute and ask directly, or you can also type in the chat. So far we don't have any questions. Maybe
I have a question.
Okay, yeah, go ahead.
Yeah, Sahil, very nice talk. So could you comment on, I know you comment on NHEJ repair. Could you also comment on MMEJ, microhomology mediated end joint repair in your work?
Yeah, so that's a really great question. Unfortunately, I do not have experience with that, but I've read publications where people are using basically just, these 20 base pair homology, repeats for targeted insertion. My only issue with that technique is if you can couple it with a reporter technique with high enough efficiency, right, such that you can have a readout downstream. Nevertheless, if you really optimize the design in a way that you optimize the viability, the efficiency, I think it's a good strategy to try it out, because it's pretty clean. But I do not, I don't have much experience with that. Yeah.
Okay, yeah, great. Thanks for the question. And I have one question regarding the BP53. You mentioned there that you could achieve the transient effect like 24 hours, and how to achieve this kind of effect, like just a transient?
Yeah. Good question. So basically what the single guide RNA does is that it recruits your Cas9 complex, and this Cas9 complex is coupled with this P53 dominant negative version, right? So just these, around 100 base pair amino acid. This is the region that your P53 binding protein actually uses to come and recognize the double strand break, right? So what this is doing is that it's blocking the recruitment of endogenous wild type P53, right? So this is just transient because the single guide RNA then is actually shunted out of the nucleus if you're using RNP within 6 to 8 hours. If you're using plasmid, it's outside in 48 hours. So it does not really affect the endogenous capability of the system to actually edit your nucleus or any target. So it's really recruited through the Cas9 enzyme and also shunted out through the the RNP complex of the Cas9. Just one parting remark. So so the techniques that I showed basically in the case studies, so I've tried almost all of them. I have the protocols for them. So if anybody wants really, or help also with designing, it's for free. Just basically, let me know. I like to help, and yeah, all of this material is available. Also, the field basically is exponentially growing, right? I have read papers which show actually efficiency even higher than 40 to 50% in primary cells at some stages. So it's really right about the right design and how you use the technique and tool. And yeah, like basically, Jinyu said, please do reach out either on my email, you have the slide deck, or LinkedIn profile. I'll be happy to help.
Yeah, yeah, great, wonderful.
Yeah.
If you want to try some protocol and yeah, if you need the detailed like the protocol, please reach out, and yeah, maybe you can have some collaboration also. Yeah, so thanks again for the really, really great talk, and thanks everyone for attending this webinar, and I wish you a nice day and nice evening. And
thank you, Jinyu. Thank you, everyone.
Yeah,
Bye-bye.
thank you. Bye.